Just now we invited the poster authors to have a 1-minute presentation of their poster in order to invite you to visit the poster. So if Frank Vial can come, and all the authors from the session 1 to 5, please, if you can come. So it's just few words. You are not detailing the poster. You're just introducing what it is focused on, and that's all.
So I'm Frank Weyer. I'm working for an experimentation farm and development farm in Basse Normandy, north of France. The poster I present is in fact part of the study, part of a European project focusing on sustainable production and also how to reduce inputs in vegetable crops. So the works on carrot takes place in Basse Normandy, sun flats of Créance area, and it is first based on a database built in 2005 and 2008 by the JPLM. The JPLM is one of the producer groupment in Basse Normandy. The goal of the project was to identify and explain practices having an effect on the sanitary quality of sun-grown carrots. And the database itself, which we are developing, is using information from 2,000 plots of farmlands. And will be used to try to explain about 25 variables such as yield, quality, and presently it includes around 200 parameters that have been registered in the different farmlands. Thanks to this work, we quickly saw a few parameters that were really important, that explained differences on sanitary quality and on yield, such as sowing and harvesting date, such as use of certain herbicide rates, types, stage of application. The importance also of watering application, of fertilization balance, and of fertilization frequency. So we quickly saw the necessity to understand the dynamics of fertilization in terms of the frequency and balance of the products. But the experiments we made leads to a special variability, a strong spatial variability that we cannot explain thanks to the different parameters we already know. So okay.
If you say too much, that will not visit your poster.
Okay. Thanks. And
you need suspense.
Okay, so for the suspense, we are working nowadays on other parameters directly in link with the soil, with water disponibility in soil, to explain the parts.
I'll be as quick as possible. I'm Olivier Favaron, and I run trials on carrot crop for Invenio, which is an experimental station based in the southwest of France. And today I present a poster, which is a synthesis of weed control trials. So, as you know, carrot is a slow-growing crop, and it can suffer of weed competition. And up to now, there were a decrease in the number of allowed herbicides. So, in this area, we have many problem weeds that are not controlled by the weed management strategies, and that's why Invenio, with local farmers, carried out several trials, and the poster presents efficacy results of some different active ingredients on the local flora of the Landes de Gascogne in the southwest of France. So I'll be glad to talk with you about this subject, the efficacy results, and different strategies. See you.
I am François Villeneuve from CTFN, mainly involved in carrots. I present the poster of Christian Renal. I apologize, he can't be here because he has problems. The Pilazo, it's a generic method based on two aspects. First, the levels of nitrogen in soils before cultures, and the second times during the culture quantifies the nitrogen in a pestiole. The poster presents three cards for main carrots, spring carrots, and very early carrots, and invite you to see the poster and also to see the presentations Friday in the field tour.
Hello, François Latour from CTFL. I present this poster to influence of sowing time and chilling exposure on the flower injection in carrot. For the French seed production, the technique used is seed to seed, sowing in August and harvest in August the next year. The quality of flowering depends different factors. The first genotype, after the start of development, and to finish chilling exposure. In this poster, we have compared two different genotypes. The first one is considered like sensitive to flowering. Its name of variety is Blanche à collet verte. And the second one, less sensible, is Carole. During 2 years, different sowing are compared from different seed lots coming from early or late flowering dates. Plants coming from seed from the first flowering carrot are at an earlier stage of development and with less requirement of chilling. Just one cycle is necessary to induce this difference. If it's clear that flower initiation is controlled by one major gene, it's clear also there are some additional genes involved in flower initiation.
Hello, I am Pascal Poupard from the University of Angers. The aim of our study is a collaborative project between FNAMS, GEVES, IRHS, and seed companies. The aim of this study is to characterize a fungal pathogen named Diaporté angelicae, responsible for umbel browning and stem necrosis on carrot. 3 parts are presented in the poster. One part is the characterization of a collection of isolates. The second part is the sorry, because I don't have the poster here. The second part is epidemiological study. Thank you, Mathilde. And the third part is pathogenicity testing in field and control condition. In the poster, you will see the production of the sexual stage, also perithecia, produced in control conditions. There is also an oral presentation of the global project named Diapocar, and this is this afternoon in the technical session by Julie Gombert from FNAMS. Thank you.
Good morning. So I'm presenting here this poster. It's the aim of this poster. It's in fact to present my project. In this project, we want to screen in wild relative carrot populations for polymorphisms in a single gene, and we selected one gene from the family of alternative oxidase, it's the AOX1. This gene has been linked in different plant species with responses upon different stress conditions and also with cell homeostasis establishment. So our aim is to find polymorphisms in this gene that could be later be used in breeding. So we selected 45 accessions using the Koppen-Geiger classification system and considering that the grid codes are present in different continents. At this stage, we are making the decision in each way it's better to go. If at DNA or cDNA level for the sequencing, we will use NGS platforms, but we are facing some problems in some strategies that we tried before. So after identify the polymorphisms, we will try different models to search if the polymorphisms are related with the adaptation of the plants to different environmental conditions. And we also search for polymorphisms that could be linked with differences at the functionality of the protein. And this we will make the validation using two different strategies by measuring the AOX activity at protein level and also by calorespirometry. And I forgot to say that for this we will establish in the lab the test system. This means that we will be able to really see each individual polymorphisms. Thank you.
Good morning. I'm Mathieu Jourdan from Villemorine. I'm working at the quality control at the genetic labs. And the aim of the lab is to check the genetic homogeneity of all the seed lots which are produced by Villemorine. And today these experiments are done with a field control or with the use of molecular markers called microsatellites. But in the future, we will use some new molecular markers called SNPs for single nucleotide polymorphisms. And we will present you some preliminary results of what we can do with these markers. For example, following a genetic drift during the seed multiplication or to test the homogeneity of the seed lots. So, thank you.
So, good morning. My name is Elsa Lopes. I'm from Portugal. I'm a researcher in the Institute Polytechnic of Portugal Alegre, an agrarian school. And my poster is not about carrots, but other APSs. It's about coriander. And we do the characterization and evaluation of traditional and too wild coriander from Alentejo. And the goal of the present study were studied morphological characterization of 15 traditional varieties and 2 wild coriander accessions, harvested in Alentejo, and compared to 2 commercial varieties. And also an agronomical evaluation selected by the first group of accession. Well, and the poster, it's the all result. Thank you.
Good morning. I'm Alicja Macko-Podgórny from Agricultural University of Krakow, and today I will present development of a new system for carrot genotyping using molecular markers which are based on polymorphism of insertion of short transposable elements. What's interesting about those elements is that most of them, over 50%, are localized close to carrot genes, so we were able to develop markers for introns of different carrot genes evenly distributed over each chromosome, and those markers are easy to handle, time and cost efficient, and we think that might be used for carrot breeding. And thank you.
Good morning. I'm Gabriela Machaj from University of Agriculture in Krakow, and I would like to invite you to see my poster, which is about validation and use of molecular markers for detection of cross-pollination, of carrot cross-pollination with white pollen. So we converted DART dominant markers into codominant CAPS markers and then validated them on a collection of cultivated and white carrot, and our results showed that our markers can be used as a good system for cross-pollination detection. Thank you.
Hi, my name is Mehtap Yıldız. I'm assistant professor from 100. University in Turkey. And our aim of this study investigates the genetic basis of anthocyanin accumulation in carrot roots and petiole. For that, we had pigment analysis and QTL mapping. And for the purpose of QTL analysis, a high-resolution gene-derived SNP-based linkage map of carrot was constructed with 894 markers covering 635.1 centimorgan with 1.3 centimorgan map resolution. As we had 15 significant QTL for all anthocyanin pigments, and for root type purple estimate were detected and mapped across 6 chromosomes, 8 QTL, the largest phenotypic effect mapped 2 regions of chromosome 3 with colocalized QTL for several anthocyanin glucosides and for RTP. A single dominant gene conditioning anthocyanin acylation was identified and mapped in this mapping project. And this study represents most detailed genetic analysis of carrot anthocyanin reported to date. That's all what I said. Okay. Thanks.